Anyhow, I'm delighted to introduce Dr. Roberta Raffaeta, who is an anthropologist in the Department of Cultural Heritage at the University of Bologna at present, but I think not for terribly long. She's an anthropologist. She works in medical anthropology, environmental anthropology, and anthropology of science. And she's done well, a number of things. She's done some fascinating looking work some time back on basic identity, place, and the experience of migrants in Italy from various places. But more recently and more directly relevant to the talk today, she wrote a book, which I think I won't try and pronounce in Italian. I'll just translate, which, and correct me if I go wrong, is an anthropology of microbes, and how metagenomics reconfigures the human and health. So this is the book that provides the basis for her talk today. And I also should mention that Roberta has just, I'm delighted to report, recently been awarded an ERC grant to work further in this area. Now I just stop congratulating and just become envious. She is going to take up this, this grant at the University Ca' Foscari in Venice. I shall be pleased rather than just envious, because she assures me that I can somehow get an invitation to come and visit there. So I shall look forward to that. But with that, I shall hand over. Roberta, thanks so much for being with us today. And I'm really looking forward to hearing your talk on, I should say, your talk on an ethnography of metagenomics, preliminary results and next steps.
Thank you very much. Thank you, John, and thank all Egenis for inviting me, joining you and entering into dialogue, because as I was saying it to John before, many of the scholars working there have highly inspired my research, my thinking, and so I'm really delighted and honored today to share my work with you. So I proceed to share my screen. No, wait. Okay. Can you tell me if can you see? Good. Okay.
Yes.
Good. All right. Wait, I have a problem. I cannot go on. Wait, just a second. Okay. Okay. Do you see the slide entitled background? Okay, good. So just to start, just a few words about my background. I've got my undergrad in philosophy, and I was especially interested in philosophy of science. But then, let's say for the Italian system, my major was in philosophy, but my minor was in social cultural anthropology. And I undertook philosophy studies because I was really interested in anthropology. And in Italy, that was one of the few paths to become an anthropologist. But at the same time, my take on anthropology has always been highly influenced by philosophy. And specifically, the question I'm interested at is the tension between nature and culture that is a really typical aspect of anthropological studies, and how this tension relates to health practices and knowledge. So I'm not going into detail about my background and path, but just to give you an idea of where I come from and what has influenced my research. For today, I'm going to.
Roberta,
yes.
can I just interrupt a moment? Do you want to put on slideshow, because I think it's.
Okay, because I think I am on slideshow. No.
No, we've got the thumbnails on the right.
Yes, but then before I tried to go in slideshow, but it didn't. I couldn't move forward with the slide.
Oh, okay.
Oh, if you want, I can try again.
No, if it doesn't work, this is fine, I'm sure.
Okay, sorry. So for today, I'm going to present some of the topics of the books. This book just out this spring, Anthropology of Microbes, How Metagenomics is Reconfiguring the Humans and Health. And this work derives from an ethnography that I have done in some metagenomics labs starting from 2004 and still running. In some labs in Italy, and also some field work has been done in the US, precisely in California. And also I will start from some aspects of this book to also introduce my new research, this research that has been luckily funded by ERC. The focus of today's presentation will be on some aspects that I have been mostly influenced by the writings of John Dupré, Sabina Leonelli, Maureen O'Malley, because so I have this opportunity to enter into dialogue with the authors of this work. So, and what about I found during my ethnography. So, what is metagenomics? I'm sure many of you knows, but I give a small explanation. Metagenomics is the study of the ecological communities of microbes that live in natural environments. And this discipline is within the broader umbrella of postgenomics that has halo after in the new millennium, has allowed, thanks to technological innovation, to sequence huge amount of data and to recognize that we as humans share an environment, an ecosystem of living, and that also our health is not just the property of a specific organ or of an individual body, but our health is a property emerging from an ecosystem of connection in which microbes figure as mediators between a larger environment, the environment outside the human body, and the human body. So metagenomics, thanks to sequence technology, cutting edge sequence technology, can study the entire genome of samples taken from the natural environment. And this was not possible till a few years ago, because to study a microbe, you had to isolate and to culture in vitro. So this is a strong distinction with the past. And these technological innovations intersect with intellectual climate. I'm speaking on an intellectual climate of the social sciences, in which we have these so-called symbiotic philosophies, a post-human turn, an ontological turn, multi-species ethnography. So what scientists have found, this ecosystem of microbe that is turning attention out from an anthropocentric vision of the reality into a more ecosystemic and post-human understanding of reality is blending very well with all these terms in the social sciences. And in fact, at least in the anthropological literature, in the last years, there has been a great fascination, let's say, with all the microbial discourse and with these scientific innovations. And through my ethnography, what I wanted to look at was how these philosophies of posthumans, multispecies, symbiosis, how all this was applied in the work of the scientists. Was this applied or not? And if yes, how? And that's why I started an ethnography in a metagenomics lab. I started in a metagenomic labs in the north of Italy, a very successful lab where the PI of this lab was a postdoc during the Human Microbiome Project at Harvard, and then decided to move back to Italy. So I was very lucky. And so in the book, I illustrate different aspects of the workings of scientists within this paradigm. And I confront and compare with the anthropological literature and social science literature about this shift from anthropocentric view to post-human view. But for today, especially, I have decided to focus on some observations in the book related to the tension between theory and practice, because as I said before, for those parties where I mostly found convergence, for example, with John Dupré or Sabina Leonelli's work, but also maybe I wanted to add something. So as you can see, do you hear me? Yes. I hope, yes, okay, thanks.
Yes.
Okay, thanks. So on the left, you see the traditional method, as I told you before. In the past, you should isolate, in the past, but currently also now, scientists can isolate and cultivate specific strains, species of microbes. And then they can obtain a sequence, the genome sequence of these isolated strains. But as I explained you before, the novelty of metagenomics is that from a sample, here there is the human body, but you can also take a sample from the soil or water, whatever kind of sample, from a natural sample. You can directly somehow transform this sample in DNA sequence that reflects not the isolate microbes, but reflects the metagenome. So the entire community of who is living there, and this is the phrase the scientists say. The scientists say that they study who is there and what they are doing. So they can somehow get this picture of the community directly from the sample without the need to isolate. And in the book, I try to really ethnographically really describe what happens in this shift from the sample to the reads of DNA. There are a series of passages to purify the heavy and dirty materiality of the sample, and to purify in order that the sequencing machine can read the genomic information. And then what I have observed sitting for hours and days and months and years in the lab with scientists is that those scientists don't stay in the wet lab, don't work with samples, but most of the time, they stare at the computer, they stare at these long reads, and they work on those reads to analyze them, is exactly as this woman is doing right here in this picture. And they analyze these reads through algorithms and software of, and often they also use artificial intelligence softwares to analyze this. And then they develop different graphs, different analysis of the data, especially the lab in which I did ethnography. They are specialized in constructing phylogenetic trees. So histories, stories of evolution of those microbes and their relation. So, and what I could see that in all those passages from the wet to the computational and then into the analysis, as Dupré and Leonelli has already pointed out, the scientists were theory informed, not driven. So their work was not driven by theory. So they didn't have an hypothesis beforehand well formed that they had to test to match with the sample. But instead, the theory, they had some theory that informed their work, but that was not the guiding method of what they were doing. And as Nicola is the PPI of the lab where I did ethnography, summarizes, it says 100,000 genomes can tell you more than theory. He and his colleagues have a very pragmatic approach to the work they are doing. And as Nicola, as always, took efforts to explain to me, this is also given by is especially given by the fact that their data sets are very specific data sets. They're very complex, dirty in a way. Sorry, I had a joke in my mind because many times it's dirty. Also, for example, they work a lot with fecal samples, so it's really dirty. But somehow, in all these passages that I told you before, from the in vivo, from the natural sample to the computational sequence, they try to eliminate all the dirty and complex natural materialities, but yet the sample they have derives from an assemblage of living entities, and it keeps trace of this complexity. And also the data they work with are a huge number of genomes. And also they have different scales interacting in this sample. So as Nicola said, it's very difficult to work with a theory or work with an hypothesis when you have to manage this kind of data. But during my observation, I understood that what Nicola meant by theory was different things from other scientists working with microbes were referring to as theory. For example, Nicola, sorry, I have these Italian quotes I can't translate on the spot. When I was challenging Nicola, asking him, how do you choose which algorithm to apply to your data? Because I pointed to him that each algorithm anyway embodies, includes some theory. So it's not a tool that is a neutral tool. And his answer was that, for example, it's widely accepted that the best method is the Bayesian approach, followed by parsimony and then by homology. Those three methods have mathematical properties that are totally different. But given that in our field we have some problems, given that we work with very different typologies and very different dimensions, we use the most accurate method, the one that, but the one that at the same time allow us to obtain the results in finite times. And this, in some cases, means to let 100 computers to work for 2 months. So the problem, the challenge for Nicola is to analyze his data and to arrive to a conclusion, to results, choosing an algorithm that allow him to do that. So he said to have a result in sorry for my translation, but in determined time, in a time that has an end, so that is not infinite. And therefore, even before to think, for example, biological theory or the association of this algorithm with an evolutionary theory, a biological theory, his concern is first of all with the practicality of the tools at hand. So it's first of all with a theory, but that is not biological, it's the informatic theory. And biological theory for him is secondary. So he say, if you ask me which phylogenetics theory is best, whatever answer I will give you would make very angry some purist phylogenetics that I know, who are researchers and philosophers together. And here he was ironic in his tone, you know, so like pointing to the fact that some of his colleagues, they want to over-theorize. And he continued, I mean that they have some religion fights on the field, in the field of evolution, and how we should measure it. These fights of religion, these religion fights, stays at such an abstract level and so far from the analysis that involve us that I don't even consider them. So as you can see here, Nicola and his colleagues in the lab have a very pragmatic approach. And in this approach, biological theory, which is incorporated somehow in the algorithm that he uses, is just secondary to the informatic theory that is that one that tells him what is possible and what is not at the computational level. He is very careful at describing that an algorithm is not a theory, is the computational implementation, not of a theory, but of a procedure. So again, you can see he's trying really to channel my attention to the practices of his work. And so he explained, and the fact that they cannot have a theory guiding their work, because they need to adapt the tools and the method at hand to the data, to this data that, as I explained before, are very complex, very dirty, biased, with lots of signals from very different sources, very different scales, biological scales. And so I found myself very close to what Amalia Dupré said, that in metagenomics, the status of the experimenter is assigned to nature itself, in a way, you know, with this radical bottom-up approach. And this is also very similar to the concept of Bachelard recounted by Ryenberg, that how those scientists are working guided not by theories, but by methods of knowing. Because in this working of the scientist, it's not just that there is a different priority of theory versus practice, but what I could witness is that the same concept, the same meaning of what is a theory is changing. So a theory becomes somehow a method of knowing. You know, for example, if we refer back to this idea of referring to theory as informatic theory, something that allow my task to be done in time. But also this pragmatic approach gives a different meaning to the concept of bias and error. Because given the fact that they work with data that are so dirty, so heavy, so complex, they already give for granted that the error is not something that they can take apart. Of course, they have all a series of techniques, quality check and so on, to have the minimum, to keep at minimum the error in their analysis. But anyway, they are aware that even with those techniques, their sample, their data, will contain a lot of error. As another one tell me, is here we make so many steps that we cannot quantify the error. It is not possible to statistically quantify the error. The only solution we have is that the biological results, the final biological results, have to be stronger than any bias, bias that you can have. Bias, rumore means noise, that any biological noise that you can have. So as you can see, this is a radical pragmatic approximation. So the error is included and accepted somehow. Those scientists are somehow liberated from the anxiety of having some bias or error in their sample, in their analysis, because this is not the point for them. The point for them is, amidst all these errors and noises, at least to identify a biological signal that is so strong that can emerge anyway from all these errors. And this for me was kind of enlightening, because in social sciences we are so expert at always pointing where the scientists are doing something biased, where they are biased, where there is the error. And of course there are many errors, but the fact is that they are aware and they include this error in their analysis. Exactly because there aim, their objectives is different from what a lot of social science critique assume or has assumed. And but in any case, I find interesting also to see how this scientific understanding, what happened when this scientific understanding intersects with the social political bias and theory, if we can say. As Latour, famously Latour and many others has shown, science is not immune also to other kinds of bias that are political biases. So which are the political ideological assumptions of scientists while they do their work? And in the book, I don't have time right now to go deeper, but I'm happy to answer any question case. In various chapters, starting from the second chapters where I illustrate through history, I illustrate that there were different understanding of what is a microbe. I compare the Russian vision, the Russian understanding of microbes more as an ecological community, to the Pasteurian European understanding of microbes as individual agent. And of course, already in history, those different visions are linked also to a political and social milieu that influence the kinds of sciences made. But also more recently, in other chapter, I show how pressures, for example, from the academic system or from industry in the market are punctuating, jeopardizing the work done by scientists and are transforming their practices in a way that are not so maybe so rigorous because of these pressures. And I also explain how this has given place to very famous scandals in the microbiome science. So, for example, some scientists at a certain time claimed to have found a bubonic plague in the New York metro, but this was given by some superficiality in the analysis. Or there are lots of results coming up every day in the microbiome science because it's really hot and trendy science. But also this has much to do with the academic system, the publication, the pressure to publish, the precarity also of the academic environment. And also in another chapter, I show how this metagenomic paradigm is emerging somehow as a continuation of the cybernetic paradigm of the 20th century, but is emerging in a different shape and transforming somehow the kind of reductionism of molecular biology, but still at the same time being attached to it. And again, these transformations are linked to the social and political milieu we are living right now. And I also describe the biographies of some of those scientists working in microbiome research and how they have decided to work exactly in that field, because it looks to them that working in that field is an opportunity for them to make a science that is beyond the cell, beyond the DNA, but can help them to express wider political meanings, wider political value that they feel close to them. For example, I give you some example. Pamela, with Pamela, we are discussing of the fact that scientists declare to see who is there and what they are doing, but in reality, much of what they do is to see who is there, because the technology is not yet so sophisticated to precisely see what they are doing. There are lots of methods developing to see also what they are doing, but there are great degrees of uncertainties and experimentations. So science is not yet there, exactly for the reason I said before, that the data are so complex, so huge. But Pamela tell me, the limit is in the eyes of who is looking, not in the technology. What we are seeing now are distant approximation, perhaps a page of the book, a piece of it, but you don't know if you are reading a book or notes from a book. If we want to better understand, we need to change perspective. We need to change how we look at microbes, not as object of study, but to look at how they behave and why. So there is this tension entirely in metagenomics, also in the working practices, between the what I call in the book molecular view. So as I show you before in the picture of these scientists staring at the screen for many hours, they string us the DNA. This is the molecular view. And what I call an ecosystemic view, because for its nature, metagenomics is a picture of a community of genomes. And as Doolittle and a colleague has argued, the future of metagenomics is probably playing in the tension between the study of a community of genomes or genomes of a community. So if what scientists see is just a collection of different genomes, or if it's truly an ecosystemic ecological community. And different scientists at different times of their career for different projects are trapped in one vision or in another, or in the transit between these two extremes. Another scientist, Paolo, a very peculiar character, he says, you can ask, what is the meaning of discovering all living microbes when, for example, extinction of pollinators is at its highest of the last 5 million years? But this is a problem with myself. Nobody has asked me to come here to discover microbes if I wanted to save bees. I could have gone to save bees. When I arrived here, I hoped that all would automatically be cool. But then I discovered that it's up to me to choose carefully my battle. It is not that it chooses you. So Paolo is very good at his work. He has decided to join this very successful lab. This lab is publishing the best journals in the world. But one of the reasons why he joined that lab, why he decided to study metagenomics, is exactly what I was saying before. Also this idea of working with larger questions that involve ecosystems, environment, large environments. And somehow through these words, you can also see his frustration of dreaming of saving the world, but then discovering that his daily work is staring at DNA reads for a long time. And so in the book, I describe metagenomics somehow as an aspirational technology, and a technology that helps scientists to put in relation different scales, different elements, but that for somehow it is for some parties anchored to this molecular view, to these very specific working practices on the DNA, and from another part aspires to have a bigger picture. And so we go back to this picture. And so to conclude what I learned by living with scientists is that theory is not just one thing. You know, when we speak about the tension about practice and theory, is the scientist's work more practical or more theoretical? But theory is not one thing. There are different theories. There is the informatic theories, the biological one, ontological, epistemic, ethical, cultural, sociopolitical, economic, and all those theories at the end are part of how scientists work. And so I think that the contribution we can give is to disentangle these different kinds of theory and to analyze their diversities, but especially their articulation, their interfaces, and their articulation and consequences in specific settings. So I think that just speaking about theory and practice, this tension between theory and practice can tell something, but is even more enriching if you think of both practice and theory as something that is heterogeneous and that heterogeneously interacts for specific problems and in specific settings. And so this is more or less the topic of the book. And as I told you, I took one specific part to share and to enter into dialogue with you. And I want, and almost finished, to take a little part of this book that has been then developed for the project that I will do in the next 5 years, that has been funded by ERC, that has been inspired by the work I've done for the book. So starting from the premise that humans and microbes are all entangled, my project asks, how does health come to be reconfigured through this entanglement? So this is the research question of my project that is entitled Health is Cross, and I take different case studies. For example, one is the Earth Microbiome Project. The Earth Microbiome Project wants to map the microbial diversity on Earth by crossing space, time, and species. As you can see, the Earth Microbiome Project collects microbial data from different continents. They have a huge number of samples, and then all those samples will go in one computational center where they want to analyze all the different data that derives from human samples, rocks, water, soil, different continents. So it's very varied. And through all those technology we spoke before, they want to, merging all this data, they want to develop some scenarios of health, as they say, scenarios of health for both the environment and humans. So what I want to see with my project is how in this work new ontological categories emerge. So who is the subject of health intervention when you cross all these species? And also I want to see how data travel from the partner in the global south to the computational center in the global north. And third, importantly, how all these scenarios of health in which humans and non-humans are mingled, which will be the applications in global health. So, for example, I can anticipate that much of this data will feed into precision, personalized medicine and other things. So I want to better analyze these developments. So as you can see, I explain you what I did in my ethnography and how I hope to develop in my new project. And thank you very much. I am curious to hear your comments. Thank you. All right. I hope you're still there.
Okay, sorry, yes, I am. I'm still here. Thank you very much. Sorry, I took some time to find myself. Thank you. That was absolutely fascinating, and I'm sure we have lots of questions to ask.